Thank you, Henry. So I will start out by talking about how this project got started, and at least my version of what it might be about. And through the next couple of days, we will hear from many people their parts. So almost not quite a year ago, there was a meeting of a new institute that we had started about 2 years ago. And this institute works quite differently. The idea was to bring together people from many different disciplines to think about hard problems, to think about interesting problems, problems that couldn't be done without bringing scientists, computer scientists, mathematicians together. And the first meeting of this institute was held in Snowbird, Utah, where there's many mountains. And when I was putting this together, I found pictures of Jack and Volker and Rob and others wandering around, lost on top of the mountain. I think it was Volker that was lost, actually. But in a place like this, there was a time to think about big questions. And that summer, there were 4 sessions, and the one that we started the entire first session of the entire institute was a session called Computational Methods and Terabase Metagenomics. And this was a meeting that, how many people here were actually there? Let me see, well, 3 or 4, there's some more they're on their way here. It was about 30 people, and we spent a week looking at questions of what were the prospects of large-scale sequencing in metagenomics, and what are the kinds of problems that the existence of terabase metagenomics would create, both from a scientific direction, but also from a computational, from an infrastructure, from a cost. And during that week, there was a group emerged. This is a little bit larger than the group that was there, around this idea of not just talking about individual projects, but trying to come up with some new way to think about the problem. And this was more or less the thought process that we went through. It was pretty clear to everyone that sequencing was getting cheap, right? Very cheap. In fact, so inexpensive that the costs that dominate getting sequencing done these days are really not the sequencing itself, it's everything else. So what this meant to us was that the idea of thinking about individual samples that might be sequenced very deeply, say to a terabase, would become increasingly feasible, that it was not crazy to think about terabase metagenomics. That was like the overarching idea. The second thing that came up, and there was lots of discussions about this, that there was a general, very strong interest at the meeting about diversity and trying to understand diversity, microbial diversity and microbial communities. And up until recently, most studies that tried to assess diversity were limited by the sampling depth that they could go. And it was clear that we wanted both thousands and thousands of samples, millions of samples. At the same time, we want some of them to be very, very deep, because we want to understand the diversity in single locations as well as the global diversity. So that meant that a strategy that was trying to characterize microbial life on Earth would have to be both a broad strategy and a deep strategy. We convinced ourselves, at least at that meeting, that computing wasn't really going to be the problem in the near term. Maybe it will be a problem if we can't keep scaling up computers, but the belief was that the computing contingent there was sufficiently optimistic that we would be able to handle analyzing data, at least for a while. There was a broad range of questions, scientific questions, that were being addressed by metagenomics. But what became apparent was that the community, left to its own devices, and everyone's well-meaning, would essentially start to conduct hundreds or thousands of essentially uncoordinated studies. So there's no mechanism really for coordinating across the disparate communities what people were going to do, where they were going to sample, what their protocols were going to be, and so on. At the same time, it became clear that through social networking, and this is extremely obvious over the last couple of weeks in analyzing the E. coli strain in Germany, that the global community spontaneously started analyzing the data. Once BGI, for example, put the data up last week, there was a spontaneous assemblage of a community doing the analysis. And of course, even last summer, we knew that this was very likely, that just the idea that there might be access to sequencing in a coordinated fashion, that providing the samples may not be the problem. The community might be able just to empty their freezers or coordinate their field studies. But then the real question is how to agree on protocols, how to agree that what everyone would do in some sense would make sense as a coherent whole. So it was these questions and these sort of observations that led us midway through that process to this idea of maybe a higher level concept. And this concept we called the Earth Microbiome Project, EMP, or EMP, you want to say it that way. The idea here was really quite simple. It was a community approach to systematically attack the problem of characterizing microbial life on Earth. This is a very broad statement. We're not saying anything about scale or anything here. We're trying to say there's a lot of scientific questions, open questions about what is microbial life on Earth look like, how much of it is there, how much diversity is there, how do communities work, and so on. And how will we approach that problem systematically, maybe for the first time? And the strategy, which I sort of outlined before, would be a combination of extremely deep studies, trying to probe the bottom of diversity or the extent of diversity in specific environments, but also very large horizontal surveys to try to understand how much variation in diversity is there across the different geological, geographical regions around the world. Is there really a microbial biogeography, and are there biodiversity hotspots at the microbial level? And use a combination of these two strategies, right, to refine our understanding of global microbial diversity, limits to dispersion, whether there exists a microbial biogeography, and if so, what does it look like? To get a handle on community structure and dynamics by doing time series, longitudinal studies, and some more applied questions like, could we systematically approach the question of understanding how microbes moderate nutrient cycles on the Earth, such as the carbon cycle or the phosphorus or nitrogen cycles? So there's both sort of fundamental science questions and more applied or policy-related questions that come out of this. Now, one of the first questions that we get asked when we're talking about this idea is, well, wait a second. Microbiologists are not used to thinking about their science as big science, right? Usually they work in relatively small teams, individual PI-driven labs. Are we really, trying to redefine how this science is done? Well, about 15 years ago, maybe 20 years ago, I can't remember, a long time ago, I was involved in the setting up of some of the early planning for the Sloan Digital Sky Survey. This was a venture that was started in the US, and it had some very interesting goals at the time. This was back in the 90s. There's a telescope that was built specifically to do this survey, and its goal was to map 1/4 of the sky, to go deeper in resolution and time and spectral space than any study before. It increased the volume of observations 100-fold over what existed before that time. At that time, remember in the 90s, if you talk about a terabyte, that was a big amount of data. We had disk drives then were measured in small integer numbers of gigabytes. So these were very large numbers. This 15 terabyte data set was comparable to the entire amount of information in the Library of Congress. Okay? And it was measuring the position and brightness of over 100 million objects, mostly galaxies. It was also measuring the distance by measuring redshift to over 100,000 quasars. Okay, so if you think about this, it was collecting all this spectral information about galaxies, distance measurements. It was really expanding a huge amount our understanding of the structure of the universe. Okay? And it discovered new types of objects. The resulting library that was constructed has nearly 1 billion objects that have been recognized in it. And at the time when the project was being set up, we said that the Sloan Digital Sky Survey would be a new reference point for the field. It would be a field guide, essentially, to the universe, right? And it was going to be used by scientists for decades to come. Now, this project recently ended, and it met most of its goals. It's a very large-scale project. It has thousands and thousands of users. It stood up. You can go visit it online. There's a database server. You can go query it. It pioneered workflows and computing and so on. But it didn't solve the problem. That is, there's a project being planned right now that's many times larger than this problem, than this target. It's going to be stood up in South America in a few years. It will produce probably 1,000 or 10,000 times as much data. It will go further in time. It will expand the volume another factor of 100. But the point is that this proved that it was possible to change how astronomy was being done. And it was a small number of people that organized this, about 10 people, and eventually pulled the community on board and raised money. Now, how is the EMP similar to this? Well, what do we want to do? Well, we'd like to map some amount of the microbial habitats. Problem is, we don't know how many microbial habitats they are, so I couldn't actually even write in a percentage. Maybe we'll only map 1% or 1/10%. I imagine that we will increase the volume of known diversity and known data points, whether it's sequence diversity, genes, protein diversity, by at least a factor of 100, probably much larger than that. If we only do 1,000 terabase runs, that's already a petabyte of data. That's sort of 10 times or 100 times the size of the Library of Congress, no problem. Maybe 1 million samples in the first 5 years or something. We'll probably uncover something like 100,000 new genomes from that data. We don't know, maybe more, maybe less. Probably millions of novel proteins. And if we did it,